All right, let's just dive right into this explainer. Today, we're exploring the absolutely incredible and honestly sometimes completely bizarre connections between all living things. We tend to look at the animal kingdom as something totally separate from ourselves, right? But by the time we wrap up here today, I promise you're going to see your own body in a completely different light. We are going to connect the dots from hybrid big cats to shifting genetics, all the way down to the ancient fossilized reptile remnants hiding inside your very own DNA. But to figure out how life changes over time, we first need to define what exactly separates one living thing from another. I mean, you and a dog have a lot of basic biology in common, but to understand what officially separates you, we have to look at something a bit more exotic, the liger. If you aren't familiar, a liger is the massive, real-life offspring of a male lion and a female tiger. And I mean massive. They're often larger than both of their parents combined. So the question is, if a lion and a tiger can successfully create this incredible animal, does that mean lions and tigers are actually the same species?
Well, the short answer is no, absolutely not. And the reason for that comes down to the strict biological definition of a species, which is a group of organisms that can interbreed and produce fertile offspring. So the crucial point here is that while lions and tigers can technically have a baby, the resulting liger is sterile. It can't have babies of its own. Because of this reproductive dead end, lions and tigers firmly remain entirely separate species.
And this brilliantly illustrates the two overarching types of reproductive isolation that keep species distinct. So over on the right, we have post-zygotic barriers. This means an embryo might form, but the resulting offspring, like our liger or, a mule, is sterile, or maybe it doesn't survive at all due to chromosomal mismatches. But on the left side, we have prezygotic barriers. These are the obstacles that happen before fertilization is even a possibility. Think of birds developing entirely different mating songs, or groups of the exact same animal ending up in completely different habitats, or even one group becoming nocturnal while the other stays awake during the day. They literally never cross paths, so they never mix their genes.
Okay, so moving into section 1, how new species form. Let's look at the actual mechanics of speciation to see how we get from a single group to completely isolated species.
The most classic way this happens is something called allopatric speciation, which is really just a fancy science term for speciation driven by geography. Imagine a population of animals is suddenly physically separated by, say, a new river, a mountain range, or an ocean. Over thousands of years, each group faces entirely different climates, different food sources, different predators. You get the idea. Nature just inherently favors the specific traits that help each isolated group survive. And eventually, their genetic makeup changes so much that even if you brought them back together, they couldn't interbreed. This is exactly what Charles Darwin observed with those famous finches on the Galapagos Islands. Just one ancestral species hopping to different islands and adapting into entirely different birds.
And here's where it gets wild. Speciation doesn't always need millions of years and vast geographic distances. Sometimes it happens right under our noses in the exact same location. This is sympatric speciation. Evolutionary biologists Peter and Rosemary Grant actually witnessed this firsthand on a tiny Galapagos island called Daphne Major. Back in 1981, this strange, large hybrid finch arrived on the island and mated with a local finch. Fast forward a few generations, and a brutal drought wipes out most of the birds. Only 2 descendants of that original hybrid survived. Because their mating songs and beak sizes were uniquely different, they started mating exclusively with each other. In just a few decades, right in front of the scientists' eyes, a completely isolated breeding lineage was born. Unbelievable, right?
Now, if we look under the surface of these changing beaks and different mating songs, what's really happening is a shift in genetics, specifically allele frequencies. Evolution at its core is literally just this numerical shift over generations. Take humans, for example. Look at the genotype breakdown here regarding sickle cell traits. Because the heterozygous trait, which makes up roughly 42% of this population, offers a massive survival advantage against malaria without causing severe anemia, natural selection favors it. Meanwhile, only 9% are homozygous recessive, dealing with severe anemia, and 49% are homozygous dominant, meaning they have normal cells but zero malaria protection. This shifting balance, based entirely on survival advantage, is natural selection operating at the microscopic level.
All right, section 2, the patterns of evolution. When we zoom out and look at the whole planet, these shifting genetics generally tend to follow 3 major blueprints.
First up, divergent evolution. This is when a common ancestor branches out into highly distinct species based on environmental or human pressures. Think of ancient wolves eventually branching into both tiny little corgis and giant greyhounds.
Second is convergent evolution. This is when completely unrelated species end up looking exactly the same just because they live in the same environment. Sharks are fish and dolphins are mammals, but they both have incredibly similar streamlined bodies and fins simply because the ocean demands it for survival. And finally, coevolution, where two totally different species are locked in a relationship where they actively influence each other's evolutionary path.
Now, we can easily spot whether an animal diverged from a common ancestor or converged on a similar solution just by looking at its anatomy. Homologous structures are body parts that might look different on the outside but share the exact same internal bone layout, like a human arm, a whale flipper, and a bat wing. Finding that tells us, hey, we all share a great-great-grandparent. Analogous structures, on the flip side, solve the exact same problem but are built completely differently inside. Think of a bird wing with bones versus a butterfly wing made of membranes. That is convergent evolution at work. The environment is always the ultimate sculptor.
And when species share the exact same environment really closely, we get that third pattern, coevolution. It's essentially a continuous biological arms race. Cheetahs evolved to be faster to catch gazelles, so gazelles have to evolve to be more agile to escape, which forces cheetahs to get even faster. But it's not always a predatory war. Look at hummingbirds and tube-shaped flowers. They evolved their long, perfectly matching beaks and nectar reserves together to guarantee exclusive feeding and pollination. Or, incredibly, the clownfish. It developed a highly unique mucus coating to become completely immune to the sea anemone's deadly sting, allowing them to happily live together in an amazing partnership that protects them both.
Which brings us to part 3, meet your inner reptile. We are going to zoom all the way in now to look at reptilian anatomy because the exact same evolutionary rules that shaped dolphins and hummingbirds also engineered the human body.
There's this famous 19th century anatomist, Baron George Cuvier, who boasted that you could hand him just a single tooth and he could tell you exactly what the entire animal looked like. And honestly, he was mostly right. Teeth and bones are basically durable time capsules. Deep in the Karoo Desert of South Africa, paleontologists found the fossils of these ancient, ferocious, mammal-like reptiles called gorgonopsids. Unlike standard reptiles that just had uniform peg-like teeth for swallowing things whole, these specific ancestors started developing specialized canines and incisors to chew and process food. The very reason you and I have complex rows of molars and canines today is because of these ancient predators experimenting with their bite millions of years ago.
But our connection to reptiles actually goes even deeper, literally, into the womb. Around 375 million years ago, life faced a massive problem. How do you have babies on dry land without the eggs drying out? Well, reptiles solved this by evolving the amnion sac, this watertight, fluid-filled casing around the embryo packed with a nutrient-rich yolk. Amazingly, human embryos in their first few weeks of development still produce a tiny yolk sac. We don't actually use the yolk for food anymore, obviously, but the broken, derelict genes responsible for making it are still sitting right there inside human DNA. It's like a fossilized signature of our egg-laying ancestors.
So how did our ancestors' dry, scaly skin eventually turn into soft, mammalian skin with hair and sweat? Well, it all comes down to a master gene known as EDA. Think of this gene like an anatomical origami artist. It takes simple, flat skin tissue and folds it in upon itself to create entirely new organs. In our ancient burrowing ancestors, this gene folded skin to create sensory whiskers so they could navigate in the dark. Later on, it folded skin to create sweat glands so we could regulate our body heat. It even coordinates the complex folding required to build our teeth. All of these traits, hair, sweat, specialized teeth, are essentially just modified skin organs controlled by this single ancient master gene.
And that brings us to what might be the ultimate evolutionary plot twist. Reptiles here using only one bone in their ear, right? But mammals, including humans, have 3 tiny bones that give us incredibly sensitive hearing. So where did the other 2 come from? By looking at fossil records and the embryonic development of animals like opossums, scientists discovered something totally wild. As the main jawbone of our ancient ancestors grew larger to support those complex new chewing teeth we just talked about, the smaller bones at the very back of the reptile jaw became redundant. But instead of disappearing, they slowly shrank, disconnected from the jaw entirely, and migrated up into the skull. It is absolutely mind-blowing to realize that the tiny bones you are using to hear my voice right now started out millions of years ago as the jawbones our reptilian ancestors used to chew their food.
It's just truly mind-blowing when you piece it all together. Whether we're looking at geographical boundaries creating brand new bird species, the brutal arms race between a cheetah and a gazelle, or the repurposing of ancient jawbones to give us the ability to hear music, the rules of evolution remain exactly the same. Life uses whatever is lying around to adapt, survive, and overcome. So the next time you look in the mirror, remember, you aren't just looking at a human, you are looking at an incredible, ongoing, 300-million-year-old work in progress. Which begs the question, given enough time and changing environments, what on earth might human anatomy evolve into next? Keep wondering, keep learning, and I'll catch you on the next explainer.